Cancer stem cells, or CSCs, have the capacity for long-term self-renewal and the ability to produce differentiated progeny. CSCs have been identified in a growing number of malignancies, including pancreatic adenocarcinoma, in which they can be identified based on aldehyde dehydrogenase activity and expression of cell surface antigens CD44, CD24, and CD133. Here, tumors are isolated from mice harboring human pancreatic cancer xenografts and single-cell suspensions are generated. The cells are then stained with aldefluor reagent, which identifies cells with high aldehyde dehydrogenase activity and cell surface antigens CD44 and CD24. Cancer stem cell populations are then isolated using fluorescence-activated cell sorting. The isolated cells can be used in various downstream functional and analytical assays.
The stem cell hypothesis is an emerging area in cancer biology. In essence, the hypothesis states that tumors contain distinct phenotypic and functional subsets of cells. Certain subsets of cells, termed tumor-initiating cells or cancer stem cells, can generate tumors within immunodeficient mice or colonies within in vitro clonogenic assays. More recently, cancer stem cells are also thought to play an important role in development of metastasis as well as therapeutic resistance. Originally, cancer stem cells were studied in myeloid leukemias using either peripheral blood or bone marrow samples. However, with the emergence of stem cell biology in solid tumors, a challenge has emerged to generate single-cell suspensions that can be flow sorted, then studied for downstream applications.